Welcome to Micro. All right, we're back. Unfortunately, and I mean it, I know too many of you. Entirely too many of you, particularly some of you on this side. I just saying. So don't take it the wrong way. Um, it There's going to be a lot going on. Uh, Let me take care of some of the mechanics first, and the one that came up, and we're still trying to decide. I hope I will get some information today in regards to textbook. So let me start with the syllabus, and we'll go there. For you those who don't know me, I'm Dr. Stein. This is me. Uh, my schedule's a little different. Uh, I think I tend to teach. I have less time in between classes this term. So, and certainly I can meet anytime because we can always meet virtually. But really, my only gap, and I'm pretty much going pretty much straight from 9 until 12. Then I get an hour, and on Mondays I got to go right to lab. So I'm done. Wednesdays I have a nugget of time between 12 and 3 because I finish with my 9 to 12 classes, the three in a row, and then I have like 3 hour window. That's the best time. And Fridays, I'm betting I'm out the door here by noon latest. I'm looking forward potentially to some virtual Fridays. Happy days. So I'm older. I can get away with this stuff. And moving on. So there's the email, and never use that phone number. You have my regular phone number, don't you, or don't you? I'll send it to you. Don't call, but you can text. Okay. Moving on. So this is Micro. We're using a new book, and that's what I'm getting to. So we'll start there. Uh, the book is not new to me. I've used Cowan's texts many times over the years. They use it a lot in both local community colleges. I don't know if they're still using it. At CCAC and at Westmoreland Community College, they used a lot of Cowan. Okay. And it's a very good text. It's probably more geared toward nursing students. I would think. I think they're using a different text for the bio majors. So when they say a clinical approach, that's sort of the game when it comes and I use the term all the time. When we talk clinical, it means interacting with patients. That's my long suit. The clinical aspects of microbiology, the diseases, how it causes, how it presents. Less about the classical microbiology view, which looks a lot at the intricacies of molecular aspects of microbiology, which we will cover, but we won't spend an inordinately long amount of time on because it's very technical. That's we have another class that does this that we developed called general microbiology, which is more geared to bio and pre-med majors. Okay, because in the class, typically what we find in this class, we have sort of an assortment, mostly nursing majors, and that's obviously who I teach to. And you've heard this spiel before when we did AP1. Uh, we have a few from environmental science. We have a few from other areas or disciplines that are there. Nuclear medicine will take this. Health science majors will take this, which are over in the nursing sphere for the time being. So, but again, those are typically more clinically oriented courses. The interesting thing is they were with this new way that they're doing these e-textbooks is that you're allowed that we they wanted to be incorporated in our Blackboard shell, and I couldn't find a way to do that. So they sent us saying, "Are you ready to do this?" We were, but the technology was not ready on the bookstore and perhaps on McGraw Hill's end. So that's kind of where we are. I do have a course shell if you're going to use the ebook, which you might be able to access, and I think I sent you the information. When I hear more from the bookstore, I will let you know. For the time being, the book, if you don't have it yet, it's not essential. When I always, and notice I put recommended, which means, and I think it's useful to have a textbook. It's not essential, gentlemen. It's not essential to have a textbook. Okay, we have all the PowerPoints. There are a lot. There's a ton of. I mean, Cowan's been around for a long time. The Nestor text, which we used for 1,000 years here, which I love, and we're going to use some of the PowerPoints from that, which is a little more technical, is around. I mean, if you get like a third edition of Cowan or an eighth edition of Nestor, I mean, on Amazon they'll probably pay you to take it. somebody's trying to get rid of it. Take it from somebody who's been selling on, my wife and I have been selling on Amazon for 10 years. You know, when you can get those rather inexpensively. And so it's on your budget. I realize they have whatever the plan is, that there's a different plan with the bookstore that's involved now. That's fine. If you're using it, then you should have access to this. But so hopefully that will all work. So let me know if you do have an access code, and with the information that I sent you with regard to where that shell is, where you could find it on the Connect site, which is McGraw-Hill's version of the software. But they used to provide the software 3 years ago, and then they stopped doing it, which is too bad because it made life a lot easier. But you know, it's a different world. Typically, we work on 4, sometimes 5 exams. It depends. It's very similar for those that have me in AP1. These are, again, going to be objective. Read that as multiple choice, true, false, matching type exams that we conduct here, Scantron style. Bring your number 2 or mechanical pencils, and we go from there. So that, I mean, it's test taking one-on-one. For those that haven't had me, why do I do that? Most of you are in fields that are going to require licensure, which means sooner or later you're going to have to pass a very difficult exam that's conducted in that format, objective, whether it's the NCLEX classically for most of the students or other licensure exams, you versus the computer, which brings us, we had a whole in-service, which we call convocation on Friday, which dealt a lot with cheating. I mean, it doesn't really, I don't think it applies to me so much. Every so often you get an incident where you see somebody doing stuff like that. But and I think it was more directed to folks who were online. And look, your students, I wrote the book on that. I used to take other people's tests. It was a good way to make money back in the day. That's before we had photo driver's license. Okay. They provide the photographs. Okay. I was taking calculus tests for these guys who were pre-med majors way back in 1969, 1970 at Lehigh University. And I'm happy to say I managed to get them A's in most of their classes. I won't go on the old girlfriend story where I wrote her an A+ paper and she dumped me. I know, harsh, Josh, it's harsh. See, if I say anything else, I'm going to get myself in trouble. I'll be politically incorrect. So I'm not going to do that. Nevertheless, the point is, and this is the point I made in that when we had roundtable discussions about that, you only cheat yourselves. Because sooner or later, you're going to be in an exam. Have you ever seen the way they conduct these exams now? I mean, you're in a facility with a computer. It's you versus a computer. You can take nothing in there. You're going through a metal detector. There's no phones. It's you. You get a piece of scratch paper, which they take from you as soon as you've written on it. That's the highlight film. I mean, and they kick you off as soon as you're done answering questions, and you don't even know whether you passed or failed until a couple of days later. That's the way it works. So, I mean, there's absolutely no, like, cheat now, and you end up spending untold thousands of dollars to try to get a degree that you won't be able to get because you can't get a license with that test. There's just no way to do it. It's very much different than that. Pardon me for being philosophic about it. So I don't worry about things like that in my class so much unless they become obvious. Okay. And I contributed to it once because students, everybody has backpacks and stuff like that. And I had some gal, this is about 7 years ago, over in Hale, and I didn't even look at it. The other students were sending me videos of her cheating. God bless them. I apologized to them because it was my fault. I should have been more attentive to it. It's a true story. Okay. So that's the story. We have the site that's here, this guy over here. Hello. Hi, Nicole. And it's nice to be polite. Okay. And exams, let's try not to do, if there's something comes up and there's a makeup and all the COVID and related health related stuff that you're all, I'm sure, sick and tired of after one semester about a year. So if someone comes up, let me know. If you're sick, don't show up. Go online. I record these anyway, and I'm sending them to you. And we'll go from there as I figure it out. The usual stuff that's here, blah, blah, blah. We're going to get started. So the book was the big thing. And they send us all this COVID stuff, and I have to find out whether or not I have to send. You'll probably get it 16 different times from different professors. All that related, and it's not just COVID now. And as somebody will touch on here, there's actually three that are of significance right now, and it's pertinent to our class. There's like a triple header of diseases that you start reading about. Something that usually struck the very old and particularly the very young called RSV, or respiratory syncytial virus. Very interesting virus that we'll cover. They're all viruses. Influenza, the good old flu, and then COVID and COVID-related variants. So that's part of what we touch on in the second unit. And it's just like A and P. I divide it that way as well. The first unit is the basics. It's the introductory material, which I love, that we're going to start today, the historical material. It's the sort of the anatomy and physiology portion of microbes. And then once we're done with that, then we start getting into some clinical, we do viruses in that, but we do methods of control, and we do genetics in the second unit. Then we do a lot of clinically based stuff with antibiotics and how diseases spread. And then finally, we end up really looking at the diseases systematically, meaning we'll look at respiratory, skin, genitourinary, digestive, etc. Immune system related diseases. Sounds like fun, doesn't it? Which means I'm not going to swear as much as I did last time. There's going to be a lot. Insert the word of your choice. I know what you're thinking. There's going to be I'm in a good mood. At least the Steelers are over 500. Could the Jets have played anybody but Joe Flacco? My daughter at the game was so excited. Oh, we're going to get tickets for Buffalo. Do you want to come with us? Are you kidding me? I said, Miami still has to win, has to lose. But no. So I'm in a good mood about that. All right, we're done there. Moving on. Documents. So this is it. And this is on, if you go to, and I'll flip over to just real quick over here to the PowerPoint shell, which has got to be some week one. So here's our PowerPoint shell so far. So at week one, you'll see these are the chapters that we'll be at least accessing for this first unit. Probably particularly 3 and 4 as much as anything. This outline, I'm going to show you some additional notes that I have. This is a supplemental section that I took from a different textbook, which sort of describes some of the stuff we're talking. And I'll be adding more stuff to it as time goes on. But I created I'm loading this differently than the last one because I'm not using the same textbook. So if you go from there, and you'll see that, oops. So, and I have more materials week 2, week 3, and week 4, but those are really the 4 exam units unless I choose to divide it at some junctures. So that's where you'll find this stuff. So it brings us over to the documents. And so this is the outline. So today we're going to look hopefully at just this introductory stuff and some of these historical figures and go from there. We have to look at the vital activities, the medical input, which has changed over time, and things that are associated with it. And since it's the first time I'm using this PowerPoint as sort of a reference, it'll be interesting. So we'll sort of jump around a little bit there. But the one that I hope to be able is this one with historical figures. So I'll go back to that. It saves me writing on the board. All good? Questions? Bored yet? Nobody cares about your humor. And they do learning outcomes in this. I don't know why. We're going to do that with types of microorganisms. We're going to describe their role and impact. I don't know why I need to explain the theory of evolution and why it's called a theory. Theories in science, are very close to laws. They're things that are accepted science that it's very, very difficult to dispute. There are some aspects of evolution that people will dispute, and I'm not talking about the religious aspects of it. That's a separate area entirely. But microbial evolution is fairly well diagrammed. We'll be looking at that, believe me, as we start to look into prokaryotes that are there. This is a big deal. Humans manipulating organisms. This is all GMO, genetically modified organisms. We do it The amazing thing about GMO is we got the technology from microorganisms. What's a microorganism? Okay, and it's a misnomer. The name of the course is microbiology. Micro, very very small. Basic definition: you can't see it with the naked eye. Biology, study of life. Okay, well immediately that's a misnomer because one of the most major parts of this course and another one that's significant are not life. Viruses, which are not a living thing, and something called prions, which cause mad cow disease. Okay, for one. Are neither one of those are living things by the definitions that we're going to look at. What are living things? Rule number one, they're made out of cells. These are acellular. The virus isn't a cell, it lives inside of a cell. It's what we call an obligate intracellular parasite, meaning it invades a cell, takes over the cell, hijacks the cell's machinery, and slaves it to make more viruses. That's how viruses work. And the damage they do to cells and what basically their impact on what they attach to is what causes the problems that are associated with them. Bacteria, which is the other major component of this course, they're living things, but they're only made out of one cell. And it's totally different than the cells that are inside of us. And that's really the difficult part. Most of your lives so far, when you've studied science and done cells, you've been studying cells that are like ours. Eukaryotic cells. That cell that my biology teacher got up there and went, oh, there it is. The rectangle. And the nucleus. Right? That was it. Oh, they're all like this. These are cells. Not anymore. They're not similar. They're totally dissimilar from our cells. They share some of the same properties. They have DNA, they have RNA, that kind of stuff. They got a cell membrane. But bacteria, they only got one. And the whole reason, and bacteria do one thing, and they do it very, very, very well. Reproduce. And that's how they beat us. Vast numbers. You're going to hear me say it 1,000 times. It's a numbers game. It's all about the numbers. And if you think bacteria make a lot of bacteria click, where do you see viruses? Unbelievable. I mean, bacteria just go, go, go. Viruses just do it totally differently and even more. So you're going to see a lot about that. That's really what the difficulty, I think, for students at your level is that it's not traditional anatomy and physiology. They have a lot of the same properties, but not all of the same properties. And their anatomy is very much different. We spent AP1 talking about one thing. Oh, my prescription's ready at Giant Eagle. I'm so happy. We spent the whole time talking about membrane-bound organelles in anatomy and physiology. We have a nucleus, we have the endoplasmic reticulum, we have smooth, we have rough, we have lysosomes, we have peroxisomes, Golgi apparatus, right? All those things. Bacteria got none of those. We used to say in my old neighborhood, they ain't got none. Okay? They ain't there. Okay, I know. It's not, ain't in the dictionary. I know. We used to hear that a lot. They're totally different. Where do you see it? They don't. They all have cell walls. They're all made out of one cell. And I'll go over all of these things. So there's a whole new unit of anatomy and physiology. That's right, that you have to learn. Sophia's smiling at me. Okay. Explain the ways that humans manipulate organisms for their own uses. That's the genetically modified. So what did we do? Because I lost track of that. We found that they make certain chemicals called enzymes that you're familiar with. Certain bacteria make certain enzymes, and you know what those enzymes do? That's right. They chop up DNA. So when you mix those enzymes with DNA is this giant molecule. It's enormous. That's why we couldn't study it. We could look at chromosomes called karyotyping and find some oddball things like Down syndrome, trisomy 21. But now we can look at things on a molecular basis because we could chop up that DNA into little pieces. reliably, and you can study it, you can duplicate it, you can analyze it, you can look for repeating segments. And because you can make those segments and you can utilize the technology of those enzymes and staple DNA together, you can take an organism and reprogram it. That's a genetically modified organism. We do it all the time. We insert genetic material, the COVID vaccine, whatever you think about it. And I know you all had to have it and boost it and all that stuff that goes on with it. Okay, that's an mRNA vaccine. They inserted mRNA, okay, genetically by the basis of using those enzymes to effectively recreate, make new strands of DNA that make new strands of messenger RNA, and then take that messenger RNA and put it inside of you and reprogram your cells to make a particular antibody. That's what they did. It's not new under the sun. They've been doing it for a while. That's one of the magical things. Microbes are magic because they've allowed us to study. By fragmenting the DNA, we can study it. So paternity testing, that whole 23andMe stuff, okay, ancestry doc, whatever they call it, all of that is based on that technology, okay. And you know, from a stamp, for those who've had me, you know, I've used a lot of food analogies and will continue to do so. I have a very good, I think I told you, a wonderful instant pot chili recipe. Oh, to die for. Oh, oh. Goodies. Moving on. So we'll get to the burden of human diseases. We'll differentiate the different types. We basically talk about bacteria, and it does not preclude eukaryotic microorganisms. Those are microorganisms that have the same kind of cells that we have. They cause us a lot of problems, particularly ones we have all sorts of problems with fungi that are there. They cause a lot of problems that are there, mostly problems on the skin, but still even some respiratorily. That's one area, but the area that we do see a lot of them are protozoans. The number one killer in sub-Saharan Africa, equatorial Africa, is not, most people think it's HIV/AIDS, which is all over, an amazingly large amount. Not. Okay? Malaria by a protozoan, eukaryotic cell. Spread by a vector. What's a vector? A thing that carries the disease with it. What's that vector? Okay. So it basically comes from mosquito bites. And that's not the only disease that's in that category. Malaria. Very interesting agent. Most authors will tell you, and most of us who've studied the science will tell you, the number one sexually transmitted disease by the number of people that are infected, and it continues to infect on a regular basis, human papillomavirus, HPV, venereal warts, whatever you want to call it. Okay, very, very strong association with cervical cancer and penile cancers. So a lot of genitourinary cancer association with it, and we'll get into all of that type of stuff. Number two, most people thought it was herpes. Not. Something called Trichomonas vaginalis. That's a protozoan. That spread very much in the same way. Years ago, we were talking about gonorrhea and syphilis. We'll talk about all of those. Okay. And I come from a background where those are the diseases we took. I started taking care of people with venereal diseases and things like that, sexually transmitted diseases, before the words HIV were in the lexicon. Nobody knew about it. We were seeing it, and I'll tell you the story about that, but nobody knew about it. More on that coming along. So we'll get into all that. The acellular ones, viruses and prions, which we'll talk about. Compare and contrast the relative sizes. There's different sizes. And so it deals with living things ordinarily, with the exception of those acellular forms. Remember, cell theory, all living things are made out of cells. And all basically all things come from all cells come from pre-existing cells, I guess, other than the first one. So we'll look at all of these different things that we won't pay a lot of attention to. Helminths, which are worms. I'm not that knowledgeable about it. It's not that they don't cause problems in humans particularly in this area, and it has a lot to do with our water supply. It's much more of a problem. We have the great and very, very good fortune of having a fairly pristine water supply. Most of us do. That's a big issue. Big issue. It's there. So I don't. I'll comment on them. Technically, they form worms which are macrobial, so you can see them, and that's another reason why I really don't touch on it. Algae is very significant. Fungi, and we deal more with yeasts and yeast-related infections than anything else. And archaea because we don't really see them in the environment we're in, but we'll have to comment on those as well. Extraordinary. They're dedicated. They've got to do nothing else but reproduce. They don't generate a great deal of energy, but they reproduce very simply. They divide in two. It's called binary fission. No mitosis, no meiosis if we're dealing with bacteria. That requires paired chromosomes. They don't have them. We'll get into all of that. The advantage we have of why we use them so much in the laboratory years ago. They used to do, and it's frowned on now. When you started to develop technology like antibiotics or antivirals, you would test them on living things. They used a lot of animals. And I can give you more stories about that from my background that aren't particularly pleasant because we did a lot of live animal experiments. It's not fun. It's awful. As I reflect back on it, given what, and this is 50 years ago, that was SOP, standard practice in medical school. Now we're able to expose microorganisms to these agents in the laboratory and get a pretty good idea of how they're going to work because we work the same way for the most part. They have DNA, they have RNA, they have cell membranes, they make proteins, okay? They excrete things, they ingest things, very, very similar to the way we do business that are there. Obviously, you can't see them. We're going to get to the microscope part, and I hope relatively quickly, okay? A lot of times, we examine them indirectly. For many of you going into clinical fields, that's in the hospital. What does it mean to analyze them indirectly? How do we know if someone how do you know if you have COVID? You take a COVID test. That's an indirect means. Okay? You're measuring the antigen. You're measuring in, let's say, a nasal secretion like mucus, and you put it in one of those little systems everybody's seen, and a little line comes up. Okay, that's technically an indirect means. This is not. It's very. Most of us don't have the capability of having an electron microscope in the house. We don't have one here at the university, and yet we use microscopy all the time. So when we talk about a microbe, it's got to be something. See, mushrooms you can see with the naked eye. So by definition, they're not microbes that are there. And we'll get into the historical stuff. And for the time being, let me shift gears. I'll get back there. And I want to do the historical figures. So, how did it all start? I mean, historically, for you and I, we know about things, particularly in the COVID era. We're very, very well aware of if somebody starts to cough or sneeze around you now, we're very, very sensitive to that and go, oh boy, I wonder if they're getting sick. Something's going on. Do I want to be in the room with them, or do they want them coming into a room and I don't want to get sick? We're much more familiar with that because of, and maybe that's the best thing that's happened. We've become more cognizant of how diseases are spread, particularly those that are spread by droplets or respiratorily spread, as that one is. But historically, there wasn't a lot done about that. It's interesting. Early on, okay, in the early days of medicine, we're talking about ancient Greece and ancient Rome, there was an understanding that there were things that we couldn't see that were causing diseases. Okay? It came from the food, or it came from the water, or it came from being injured by something. Maybe there was something on the rock or the sword or whatever, the arrow that might have been involved. That's kind of, so there was a general understanding. A lot of the things that we use in medicine were developed by the medicus, which were not very well respected, but they were the precursors of the modern physician, who took care of the gladiators, who sewed them up, who cauterized their wounds, all that type, and they basically treated them not a whole lot different than we treat today, with a variety of different herbs and materials and things like that. And that's how they studied how their effects. That was really the foundation of pharmacology. I mean, aspirin comes from tree bark. Let's be clear about where those things are. Bacteria, antibiotics, just like bacteria, come from mold. Antibiotics are made by other microbes, by microbial forms. That's the definition of an antibiotic. So they knew to put certain things on that were going to be advantageous or stop the bleeding, things like that. And then what happened at the fall of Rome, we had an area called the Dark Ages. And the Dark Ages took place until roughly the Renaissance. And it was over, and this is not a knock on the church or anything like that, but it was basically, at least in Western medicine, and I'm not that familiar with Eastern medicine, with Asian medicine, there was a very, very limited understanding. The feeling was that when people got sick, it was they were invaded by the devil. That was certainly true for mental illness. Something it was a punishment of things like that. If the arm was damaged and infected, you cut off the arm. Those were the treatments. It was really, and a lot of times it was representatives of the churches who were making the call on that, and you were putting yourself in jeopardy to go against it, until a time called the Renaissance, when people began to rethink about things. If you've ever wondered about, how Michelangelo and Da Vinci were able to do some magnificent sculptures and art and reflect the body so brilliantly, and some of the ancient anatomical, it came from them cutting up dead bodies, which you couldn't do. They were doing it in areas where no one would observe them, so they could learn about it. That was sort of because you were at peril. Those things. I won't even go into things about Galileo and Copernicus and all of that kind of stuff. It's fascinating historically, is what I'm telling you, and that's where these historical figures were very, very interesting, because they were constantly they were challenging this notion. So I wanted nice face. Gilly. This was the theory. Abiogenesis literally means life from lifelessness. Now, we all know living things come from other living things. Cell theory 101. All cells come from pre-existing cells. All organisms come from other organisms. This is like, boom, that chair is going to come alive, which I think sometimes it does. Thank you. One person smiled and hold. You know how I respond to my bad jokes not being received. It's early, I know. For those of you who used to, it was 8 o'clock, I could accept it. 9 o'clock, not anymore. Abiogenesis, life from lifelessness. So the concept was also known as this, spontaneous generation. That magically something would be infected, that magically something would grow. And so it became an effort of science, as science began to become sort of recreated, if you will, in that time of the Renaissance, and so they used scientific theory, which you're supposed to know about. Okay? What's scientific theory? We observe something. Okay? From an observation, you get an idea. If I'm watching my students and they're sitting there like that, I'm not doing anything very interesting. That's my observation. I better change something and maybe speak louder, try to crack a joke, do something silly. Okay? Maybe that'll wake them up. Okay? And if it does, so that's my hypothesis. I experiment doing that. And if it succeeds, then to some extent, I've proven my hypothesis. That's what this is all about. That's where this comes from. So we're going to look at, and I try to put them in some way, shape, or form chronologically. So early in the 1660s, an Italian biologist who studied living things named Reddy, I don't recall his first name, and depending, and you can, and there'll be all sorts of illustrations online and some controversy as to how he did this. What he did was he observed that when meat was left out, it's not like they had a fridge that was handy, and the flies would like the meat would stay there. Yeah, it would turn colors, but once the flies got on it, all of a sudden, shortly thereafter, you'd start to see little maggots, little wormy things crawling out of it. Okay? He reasoned that the flies were laying eggs. So he conducted an experiment. He said, I'm going to put them in basically a glass petri dish, a little glass container, and I'm going to have, and he actually had three of them. He had one that was covered. So air, remember the evil force was in the air, was the theory. That spontaneous, some life force was in the air. And with it covered, the air couldn't get to it. Well, obviously, it didn't demonstrate any signs of maggots forming because the flies couldn't lay their eggs on it. If you left one open, flies had a great time with that. How do they come? The flies would light on something that very cute dog of mine would leave someplace and get that on their little appendages, and they lay on the meat, and the next thing you know, voila, microorganisms would start to grow and go into larger organisms that we could see. Then he used cheesecloth, basically a very fine mesh gauze, and he did that. That way air could get through, but he reasoned that the flies laying their eggs could not. And he was absolutely right. His theory was there, but everybody started, oh, there's something in that life force is being repelled by the cloth. And depending on what cloth that they used, they couldn't validate the experiment. How do you validate? You try to replicate the conditions and get the same results, even if you're someone who doesn't believe it. We do that in science all the time. Oh, so and so did an experiment. Someone on the other side of the world or the other side of the university will try to do the same experiment and see if they get the same results. That's a process of validation. Okay, you test and you retest because we doubt like crazy. Everybody's trying to poke holes. Sometimes because they just like it's their nature, or sometimes that's really what science is about. So he was the first one. So there was it was somewhat controversial. Thankfully, he managed to survive whatever inquisition was made of him trying to disprove church doctrine. Now, not at the same time, but somewhat later, and there was other people trying to do the experiment. An interesting fellow named Edward Jenner in the 1790s, he addressed a disease that killed untold millions of people because we don't have an adequate count. The two big diseases were plague, which is insect borne, carried by fleas. Okay, and we'll get into the way that is, why rat control was so effective with regard to it. And the other, smallpox. His was smallpox. He made an observation, very famous. He saw milkmaids. Milkmaids would get on their hands a topical infection called cowpox. These days you're hearing about monkeypox. Same family of viruses. They got the cow, and he said, they never get smallpox. I wonder if there's something in those cowpox that I could pull the fluid out of. Pox is a pustule sore. And if I injected that into somebody who didn't have smallpox, waited till it worked, whatever that meant, and then exposed them to smallpox, and they didn't get it, I'd have a way to treat it, prevent it. Preventative medicine, what a concept. It worked. Now imagine, and this is what scientists did. They did it on themselves, they did it on their families, they did it on volunteers. Because they knew if they, took someone who, a prominent person, and it didn't work, they were going to be executed. That was in around 1791 or 1793, I don't recall. And 180 years later, as of 1977, we have not seen any case of smallpox. It's been eradicated. He developed the first immunization, and we talk a lot about immunizations today. Another name for immunization, as everybody knows, is vaccine. Why do we use that word? That part. Vacca, the Italian word root for cows. And I thought I was being clever. You probably heard that. Moving on, historically speaking. And see, I'm not getting any real bombs out there today. Semmelweis, puerperal sepsis, childbirth fever, midwives versus doctors. Before Pasteur, we probably won't get to Pasteur today, or at least we'll maybe touch on it. Before Pasteur did the landmark work that really refuted spontaneous generation or abiogenesis. Before that occurred, Semmelweis made an observation, again, scientific method. He saw medical students and doctors coming from the laboratory. It's not the lab that you're going to be doing micro in. Okay? The lab was where they cut up dead bodies. They had duck all over them. You were happy if they wore an apron other than their clothes. Their gloves were rubber, they were covered. And they'd go up and deliver babies. So imagine how easy it would be to transmit a disease. Didn't wash their hands, didn't do anything like that. He looked at the rates between doctors and midwives. Midwives were much more cleanliness in the process. And he said, I'm going to start having the medical student, because he was the boss of the hospital. He was the lead physician in Vienna. Vienna was like playing the palace. Okay? Vienna was Mozart, Beethoven, they all played for the emperor in Vienna. Vienna was it. That was the upper echelon. So he wrote a paper about that when it was proven that he reduced the rate substantially. You would think he would be celebrated. He was ostracized because he wrote the first paper that pointed the finger at the doctors. And the other doctors didn't want anything to do with him. So they kicked him out. Interesting, he died of an infection some years later. Just to get, as I'm fond of saying, and I say it all the time at home, no good deed goes unpunished. You ever hear that expression? I live by that. Just letting you know. It's the way it works. Be careful. So that was Semmelweis. And so there was a problem there. And about 10 or 15 years later, we will get at least to the start of Pasteur. Pasteur came around, and Pasteur, he already had credibility. His credibility came from the wine. Now, Pasteur was a scientist. He used the microscope all the time. Oh, I forgot about the microscope. You didn't remind me. Historically, because the other PowerPoint sequence has it, the microscope was created, okay, by a guy named von Leeuwenhoek, who's not in here, but undoubtedly you'll see, don't mention it. And von Leeuwenhoek was not a scientist at all. He was a fabric merchant. And what he did, he used to use magnifying glasses to look at fabric. But when he traveled, he was either on a boat or crossing a lake, or even on the ocean, and he developed a device. It was a single lens microscope that could magnify 300 times, and I'll show you a slide next time we meet because I don't have it primed and we used to make it in science class. And his original drawings will look just like things that you will see under the microscope in laboratory. The elementary bacterial forms, which you can see they were magnifying about 300 times. You're going to magnify it more than that, up to 1,000 times. The first compound microscope, when you compound, it means you have two lenses optically set up that one kind of magnifies the other. So you have 10x and 10x, and it'll be 100, or 10x and 40x, it'll be 400. That's high power. Or 10x and 100x, which is oil immersion, which you're going to use more in the laboratory. Another scientist put that together. His name escapes me for the time, but also they were the ones who really started with it. So Pasteur was using a compound microscope, not unlike what you're going to use in the laboratory. And he had credibility. What he did, the wine industry was suffering in France. Their wine was spoiling. And they imported and exported wine all over the world. It was very, very popular. So they were losing a lot of money. And they said, can you figure this out? Well, he took samples and he found that there was a lot of yeast in the wine excessively. They used yeast was a natural was really caused the fermentation. And what he found was that the that the yeast, if there's nothing done to check its activity, would eventually the wine would spoil. And so what the wine would spoil, what happens to alcohol when it's exposed to yeast over time, it changes. It basically produces acetic acid. Acetic acid is the foundation of something we have almost in all of our cupboards in the kitchen at home called vinegar. Here it gets that Latin word root, vin for wine, agre for sour. Vinegar means sour wine. Now, where it may be, maybe you have a nice quality balsamic in your pantry. We do. Okay. We also have sherry vinegar, very, very useful when you're cooking Spanish cuisine, that are there, and good old white distilled vinegar. So what did he do? I got to figure out a way to stop that from growing. So what he did was he developed a system. So he started heating the wine. If he boiled it, it didn't spoil, but it had no flavor. So he said, if I heat it to different levels and just hold the heat there with a thermometer, and he came up with this one, roughly at about 62.5 degrees Celsius, which is about 140 degrees on our Fahrenheit scale. He held it there for 30 minutes. The wine would still hold its flavor and it wouldn't spoil. So we gave it a name. Or we gave it a name, I don't know if he did. Think of your dishwasher. Internal temperature of a dishwasher is approximately 140 degrees Fahrenheit. How long does it hold that temperature? So when you're washing dishes in the dishwasher, effectively, what that does is it rids it of most microorganisms that would spoil and typically cause digestive disease, things like E. coli, which are classically from things that are soiled or found on soiled hands, things like that, that potentially could cause significant illness. That was how he started. So Pasteur, starting like that, what he was able to do over time was modify that, and we'll go into the method of pasteurization when we look at methods of control. And because he had that credibility, they said, we've got to find a way to be able to determine whether or not this idea of abiogenesis or spontaneous generation, how we can disprove it. Because by this time, scientists were pretty sure microbes. And what we'll get into next time we meet is we'll look at the rest of this and talk about the microbes. So thank you very much. I hope you had a good first day here. See all of you soon. Hopefully this is still recording. It looks like it is. Bye.